Standardization of the data collection, the data processing and curation, and the models that we're using, those are all very standardizable and really help build that institutional knowledge.
Welcome to Data and Biotech, a podcast from Cordi where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Wesley Tatum, welcome to the Data and Biotech podcast.
Hey, thanks for having me. It's good to be here.
Awesome. Well, just to kick us off, would you mind giving us an introduction to your background and what brought you here?
Yeah. So I'm a principal engineer at Serán BioScience. We're a CDMO out in Bend, Oregon. And over there, I'm leading a small team of scientists. We're evaluating different compounds. We're figuring out either limitations in bioavailability or in manufacturability. My team helps figure out what the problems are, what the solutions could be. We screen the solutions, help select the lead, and scale up the processes.
I think it would help. So if you just outlined like why biotech organizations come to a CDMO like Serán, and where you meet them when they come to you, and then how your work kind of goes from there.
Yeah, so there's a lot of different points in the process of drug development that they can come to us, but by and large they come to us because we have an expertise in formulation. So formulation and manufacturing are kind of the last two bits before you're putting drugs into people. So they're coming to us, let's say, on the early end, it could be discovery. They have a couple of different candidate molecules, and they want to compare them, try to understand what's going to have the best exposure, what's going to be have the most stability, the most manufacturability. We help them, really early in preclinical studies, narrow down those compounds. Maybe they already have a compound lead that's selected, and they need help with GLP tox manufacturing. We can help them with that. Or they've already gone through GLP tox and they need to develop a first-in-human formulation. A lot of your tox formulations are suspensions and solutions, and typically you're wanting an oral solid dosage form, whether that's a tablet or a capsule. And so we can take their drug and do that and compare the exposure back to their GLP tox exposures. Or, on the further end of things, maybe they've already gone through phase 1, phase 2. They were maybe, pushing through a capsule, like a powder and capsule type formulation, and they needed something that's a lot more scalable. And so that's the other thing that we'll do is we'll take in, phase 1, phase 2 programs that need to be reformulated for either bioavailability or manufacturability. So kind of all along the, that development roadmap, they can come to us because of our expertise.
Yeah. And, when we talked prior to the podcast, you all mentioned that Serán specializes in difficult drug products. So, would you mind just telling us like what are the kinds of challenges that make a drug product difficult? And then how does that alter the approach that Serán takes when working with organizations that have difficult drug products?
Yeah. So, I think the fun part of it is that difficult drug products are difficult for any number of reasons, right? We're especially with the new molecular space, the compounds are lower solubility. They're more lipophilic, but not lipophilic enough to enable, say, a liquid-filled capsule or something. So maybe it's just solubility, or maybe it's permeability. Really, they're coming to us because we like to take a technology agnostic approach. We do a lot of spray-dried dispersions, but that's by no means the only thing we're going to try. We're going to try, HME is another option, or just a conventional crystalline tablet formulation with some, maybe micronization, or maybe you need an acidulant or precipitation enhancers. So really what we're doing, and I think our expertise, is trying to understand really what's limiting the molecule and how we can come to a robust process that's scale up.
Yeah. No, that makes a lot of sense. So what would interest me is an organization comes to you with a new molecule and a new compound, and they want to understand from you, how to improve the solubility or the permeability or the manufacturability of the compound that they're bringing in. And so I'm interested in what types of data do you collect, or how do you characterize where you take the conversation from there?
Yeah. So especially if they don't have any data that we can leverage, or, I guess another really common problem statement is they had all this data when they were first developing the drug substance, and now they have a GMP drug substance manufacturing process. All of a sudden, they've reduced their impurities. They're getting different crystal forms or higher levels of crystallinity, and all that data might be, totally unrepresentative of the material that we have to work with. So the big data that we want to take to understand what the problems are going to be are solubilities. So crystalline solubility in different biorelevant media. So that's fasted state gastric fluid, fasted state intestinal fluid, the base buffers that go into those. So we're really trying to understand how the drug is going to be thermodynamically soluble throughout the GI tract. And then we also want to understand the amorphous solubility, right? If solubility is going to be the limiting factor here, we want to understand how much of a gain just going to the amorphous form can get you. So, we have a couple of experimental protocols to be able to take those amorphous solubilities. And then we also, if we or they or us together have identified that it needs to be amorphous, we also have some screening methods that we can use for looking at different polymers that it might be most compatible with. So solubility is really important, but we also want to understand a little bit about the nature of the molecule and the crystalline drug substance itself. So melt quench is a really common technique on differential scanning calorimetry. So essentially you take your crystalline material, you heat it up until it melts, and you quick quench it down and capture it in the amorphous form. And then you heat it up again, and you can look at the melting point versus the class transition temperature. Where those are individually can tell you a little bit about physical stability, different types of processes you want to turn to. But then the TM over TG, that ratio, also gives you insight into the thermodynamic drive for crystallization. So if we are going to do an amorphous form, how much are we going to be able to push the drug loading, which might be purely a factor of how stable we can get it versus performance. That's kind of the main thing, is what are the physical stability risks and what's going to be the tendency to recrystallize.
So you mentioned solubility and you mentioned amorphous forms. Would you mind just, introducing people to amorphous solid dispersions and why amorphous solid dispersions, help with solubility, and sort of what you're sort of driving at with the experiments that you're doing in order to characterize whether that's the right thing or whether there are other formulations that are going to be more appropriate?
Yeah, absolutely. So I guess, just starting with your first question, why an amorphous form and what is it? So when you have your crystalline structure, right, there's kind of two steps that it takes to solubilize a drug. First is your enthalpy of sublimation, essentially. So you have to break that molecule out of the crystal lattice, and there's a energy to do so. And then there's an entropy of mixing. You have that drug, and now you need to get it off and be free drug before it, because it wants to be in that more stable state of being in the crystal lattice. So what an amorphous system does is it reduces that first step, right? So now all of a sudden you don't have these really stable crystalline structures, and you have hydrogen bonds, maybe dipole-dipole interactions, all these positive interactions that make crystal lattices so stable, and you disrupt those. So now maybe you only have some local interaction between them, and it's a lot lower energy cost to be able to remove that drug from the whole solid, and then your entropy of mixing is going to be the same, right? So you're reducing energetically how much it takes to solubilize the drug. And so therefore there's a higher thermodynamic drive to have drug in solution. So your crystalline solubility is always going to be lower than your amorphous solubility. And when I'm talking about solubilities here, I'm referring to only free drug, not drug in micelles or drug in polymer colloids, which is, another aspect of amorphous dispersions. So that's just, if we're looking at the crystalline drug and versus the amorphous drug, that's kind of the basis there. Now, an amorphous solid dispersion is then when you take a polymer and you mix it in, and that's going to further separate the molecules, and it's going to help make them a little more stable. Because if you just have the amorphous drug itself, it's going to want to tend to go back to that crystalline state, whether it's, you're sitting in your GMP stability experiment at 4075 and you've got heat, you've got humidity, and it's always going to want to go back to that crystalline state. So you can stabilize it by dissolving in a polymer there. And then the polymers also have implications around bioavailability as well, but that's a different aspect of it.
No, I'm really interested. And if you can connect sort of like the formulation that you're doing in terms of like how your clients are thinking about dosing or thinking about, the way that the drug is metabolized by patients. Like I think that like that entire sort of logic cycle is really interesting if you're able to spend some time.
Happy to do so. So in addition to having higher free drug just because you have the amorphous form, you are also able to form drug polymer colloids. Now there's a lot of really good literature out there, some really good research being done on how those form, why they're bioavailable. Kind of the high level summary is you have, drug in colloids, and those colloids themselves are able to also diffuse. So, we're actually presenting some research this week that characterizes some of the different colloidal properties. So Z average, hydrodynamic radius. We can also get at density through centrifugation. And we're trying to understand how different polymers, different drug loadings, how those different colloids differ. Regardless of how they differ, what they're going to do is they're going to diffuse. They're able to also diffuse through the unstirred water layer. That can be really limiting for some drugs, that especially if they're not as lipophilic, they're just hydrophobic. So they're going to get to the unstirred water layer, and they're going to take a really long time to diffuse through that to get to your intestinal wall. But you can have drug polymer colloids acting as a shuttle. So they'll go across the unstirred water layer. They're going to just hang out there at the intestinal wall. And as your drug gets absorbed, you have free drug right there ready to resupply it. So rapid resupply is kind of how we refer to it. And it can be incredibly enabling for bioavailability.
Yeah, interesting. It's like there's sort of like the goal, if I'm understanding correctly, is to make it so that the drug is being taken up by the patient's system, sort of like at the right rate and in a predictable way. And, make it so that it's something that can be manufactured easily and at its scale. And these are sort of like the different components that you're trying to like balance as you're doing these kinds of experiments.
Exactly. Right. Yeah. And I think balance is the key word there. When we're just doing our screening studies, we don't want to just maximize performance at the cost of stability or at the cost of downstream manufacturability, right? The polymers that you choose will have different hygroscopicity is a big one. So PBPVA, HPMC, E3 are really common amorphous solid dispersion polymers, but they're really hygroscopic. So you can fit less of the ASD into the tablet. They're going to have different stability issues compared to maybe an enteric polymer like HPMCAS or Oidrigate. Those are going to be able to enable higher ASD loading into the tablet, and they'll have less water uptake. But then you also have to balance stability risks because they have a lot of acid content in them. So if you have a drug that degrades in the presence of acid, it all goes back to balance. What's going to be best for all of these different pillars of formulation, not just bioavailability.
You mentioned rate limiting steps. Like you're trying to understand like what's sort of blocking the drugs, the drug moving forward. Is it performance or is it manufacturability? And then like what are the issues with performance or manufacturability that you have to sort of resolve in the process of developing the formulation or the way that it's being manufactured? Am I thinking about that right?
No, that's exactly right. Yeah, you got it.
So you've got all of these ideas, some of which you've just shared, of like sort of the decision tree of like potential formulation decisions that you could make, or different polymers, different excipients that you can bring in to resolve different issues with the drug and make sure that it's that it's manufacturable, that it's soluble, that it's stable. How do you narrow the process? How do you determine like what experiments you need to run, or how you get to the point where you can then make a proposal to a customer about like the appropriate formulation?
Yeah. So we actually do something called a developability assessment, where we're taking that solubility data and thermal data and we plug it into a couple of different models. So, BCS, DCS, those are fairly well known, biopharmaceutical classification system, developability classification system. And we're just trying to bend these different problem statements. So how much are we focused on just solubility? How much can we get away with improving just dissolution rate, right? So the difference between DCS 2A and 2B is you low solubility, but in 2A, if you just help it dissolve faster, you'll be fine. You've got high enough permeability. So that's going to kind of drive which technologies we might evaluate. We're also looking at the thermodynamic rate limiting steps for bioavailability. So Kiyohiko Sugano's put together a really good framework called the fraction absorbed classification system, where you are looking at those 3 rate limiting steps. So dissolution, solubility, and permeability. And just by looking at the molecular properties, some of these thermodynamic measurements that we're making, we're trying to understand how much are we going to be solubility limited versus permeability limited. And then one step further, how much of that is at the unstirred water layer boundary versus at the intestinal epithelium boundary. Kind of the first thing we do is we take those measurements, we put it through these different models, and we understand what technologies are even worth exploring. We're not going to just say, oh, micronization will help if you're BCS class 4 or 2B. Clearly, you need some additional solubility enhancement. So there is that broad decision tree, and mostly what we're trying to do is prune off forks of that. And, each of those decision trees have, a fractal amount of other variations in them.
What are the conversation points like with your customers as you're going through this process? How do you sort of communicate how that decision tree is evolving, or like which aspects you're thinking about pruning, and like how do you gather input from your customers about what things you should try or not try?
Yeah. I mean, it is constant conversation, and that's something that we also really like to pride ourselves on. Is we're not just taking their molecule and running with it. It's their molecule, and we're just a part of the team. So we're meeting with them weekly, bi-weekly. Maybe we're in the middle of something and we get some really interesting data or some really concerning data. We want to take that to them immediately, talk through why we think it's promising, why it's limiting, and possible ways around that. So, the developability assessment, the report, that provides a really good framework for where we think the just thermodynamic limitations are. And we can kind of give caveats to each of those, right? Like, oh, this says you won't get this, but really there's all these other factors to consider. So this might still be a beneficial avenue to pursue, or no, this seems right out. I don't think we can pursue that. Maybe they still want to pursue it, so we'll give it our best effort. But we're trying to take into account, what kind of manufacturing processes that they would like to pursue. So maybe they really don't want to do spray drying for one reason or another. There's more operations, you're using solvents, and they really want to focus on HME. Okay, well, how's their thermal stability? How strong are the interactions with the polymers? Is that a feasible route? And, do they have the material for us to look at an HME? You need at least a 0.5 kilogram of API to get on something at scale. There's smaller things you can do, but there are material considerations. So we're balancing, maybe they only have 1 gram of material to work with. Okay, well, let's evaluate these avenues first, and then we can come back and reevaluate these other avenues later. Or maybe they have a really high dose, right? Like with some of these new compounds, it's not unheard of to have 1 gram or more of active that you need to dose in a day. And that's really going to drive what sorts of technologies you think are feasible.
Yeah, it's interesting. And so you're sort of helping them navigate like the scientific constraints of the problem, while also sort of sussing out the economic constraints and the supply chain constraints and the manufacturability, the logistical constraints, the engineering constraints that are on the problem at large. So like you can control you can communicate sort of like the scientific and physical and chemical nature of the way that the drug needs to be managed. Like you also have to track like all of the external things that lead to the drug being appropriately manufactured.
Exactly. Yeah. There's a lot of just practical constraints where, and let's say we're doing a formulation screen and we have one polymer that just knocks it out of the park. We actually have this quite often where a 25% drug load in PBPVA looks really good in vivo and in vitro. And let's say maybe HPMCAS it's good. It's better than the amorphous form, better than the crystalline form. But it's maybe 30% less good than the PBPVA formulation. And the client might say, okay, we have to do this PBPVA formulation. That's going to be our one. Well, we can take them through the math and show them that, okay, well, we can fit 30% more of the HPMCAS STD into a tablet. And so really the bioavailability all levels out. So it's taking those practical constraints and all those downstream considerations and trying to think of them as early as possible so that we can make the right decisions at the start. We don't end up painting ourselves into a corner.
Yeah, that makes a lot of sense, and it's also the point where picking the right partner early on that can take you through the entire journey helps you to make good decisions early on that like lead you to like better situations later on.
Exactly. Yeah, we have a non-zero amount of what we call rescue programs where there was a formulation that was landed on for maybe phase 1, and it's the client recognizes it's not scalable. And they come to us and, we're trying to figure out, okay, based on the data based on how they got there and all the data that supported that, what can we figure out? What additional data do we need to be generating to come to a more robust process?
Yeah. So each of these screens that you're talking about, or assessments that you're talking about, I'm interested in like, how standardized is the process for each of these, and how much of this is you sort of like coming up with methodologies like on the fly, given the tools that are in your toolkit, and then like are there consistent like computational approaches that you're using that kind of scaffold these things and allow you to like make them more repeatable or like make them more efficient, that sort of thing?
Yeah, no, that's a great question. I would say it gets more tailored to the compound as you go through the process. So when we don't know anything about the compound, we got to get the basic data: DSC, PXRD, aqueous solubility as a crystal component, aqueous solubility as an amorphous compound. So those are pretty standardized. I mean, the gastrointestinal tract's going to be pretty consistent. So we're always looking at those. Now, the big thing is that we're trying to make sure our experimental protocols are also very standardized, because some of these compounds can be incredibly sensitive to ionic strength. And so if you have one analyst that has to, they overshot the pH of the media and they had to titrate it back down, and your ionic strength is, instead of 28 millimolar for BASF V1, you're looking at more like a 35. Maybe it doesn't matter, or maybe it really does. And you don't know, and you don't know what you don't know is the big thing. So we're really trying to have standardized experimental protocols so that we can tailor what we're actually putting into those experiments to the compound itself.
Yeah, and I'm interested in like, moving from like compound to compound, from customer to customer, like how transferable is the knowledge that you get? Is the extent of the transferability like just the experience of going through the process multiple times with multiple customers, or are there aspects of experimental design or of data gathering or of computational approaches or of standard operating procedures or of just the way that you work with the compounds that you're receiving? Is that like standardizable or transferable for the organization's knowledge to like grow over time?
Yeah. So I guess I'll first say that even within a molecular series, so let's say a client brings us multiple compounds, you'd be surprised how often a methyl group or changing the site of your chlorination can drastically change all of your molecular properties. So to some extent, the specific knowledge of classes of compounds is not that transferable. But like you're alluding to, the protocols, the pitfalls, all of that experience that you get and the problems that come up that you have to solve, those are incredibly transferable. And so standardization of the data collection, the data processing and curation, and the models that we're using, those are all very standardizable and really help build that institutional knowledge.
Are there best practices that you found for like ensuring that your team is learning, like as it's growing, is learning from the things that have come up in projects previously, or like how do you think about like embedding that knowledge in the organization as a whole?
Yeah, I mean that's some of the stuff that's hard to put into an SOP, right, or a work instruction. So what we're doing, and a big part of my role is having seen so many of these programs and having been in the lab myself, is being able to look at the data and understand where those different pitfalls might be, or how we need to adjust an experiment to be able to tailor it to the compound. And so with my team, a lot of it is training them to, by experience. Okay, we have this data, we have these trends that we're seeing. What do you think of that? Okay, and kind of just bringing them through and having them understand.
As with all science, you're always sort of up against the epistemic uncertainty of like, you're never going to have like 100% confidence of whatever conclusion you're drawing. And so, but then you're also dealing with organizations that are under time constraints, material constraints, budget constraints, in addition to like the epistemic constraints of like, how can we even know what we're trying to discover here? And so I'm just interested in how do you determine what questions need to be asked, and like, are there situations where you need to push back on the customer to say like, actually that budget is not like, we're not going to reach a level of comfort with the conclusions that we're going to be able to draw at this level of budget, or that sort of thing.
Right. Yeah. No. We have those conversations a lot, both internally and externally. And what we do is we have all of the questions, right? You can list them out. You can make a list, and then the most important thing is to rank order them and say, okay, what is critical path? What is non-critical path? What are the need to knows to advance and to hit your IND submission date or your GMP manufacture? What do we need to know to get there versus what is a nice to know that we can kind of tack in along the way? So if we're going to be doing a screening study, let's say we already have a formulation and we're about to do a demonstration batch, and we want to understand, something about the stability or different excipients. Okay, well, what of that can we tack into this work that's already there, right? So how do we make it so that it's not its own standalone experiment that's gating to the rest of the program? How much can we, find synergy in what is critical with what we want to know?
I love that answer. Do you have an example of like a company that you've worked with that came to you with a particular compound, and, like how you determined which questions you wanted to keep in and which ones you decided to leave out?
Yeah, I think probably this most commonly comes up with biorelevant dissolution. It's a fantastic tool. It can give you great mechanistic insights into things, but it's not predictive, right? It's biomimetic-ish, but you're glossing over a lot of differences between vials or USP apparatuses and the human gut. So one common thing that we get caught in is, okay, why do we see these differences, either experiment to experiment or between formulations? And it's really tempting to just deep dive into, okay, maybe there's something different in the process for the manufacturing, or, you can even get down to lot inconsistencies of excipients. And it's really easy to fall into that rabbit hole. And sometimes it's just easier to put it in vivo. And so, dissolution is cheaper and it can be quicker, and we want to leverage it as much as possible. But at the end of the day, you can get a concrete answer by putting it into an in vivo study. So that's probably the most common thing that happens. And then I'm trying to think about an example of synergy that we can find. So one of the problems that I alluded to is drug substances are evolving as we're trying to develop drug products. So we're doing a manufacture with like maybe a med chem lot, it's only a couple grams, maybe only a couple 100 milligrams. And, we can time things, or, if the timing works out and they get new drug substance, we can run that in as a control, right? So whenever we're doing biorelevant dissolution, there can be variability from experiment to experiment. So we like to always include a crystalline API control, so we can see, just a sanity check. Did we make the media right? Did we forget to turn on the heater? Or are we stirring at the same speed? All of those can kind of have effects on the curves that you're seeing. And so we have our crystalline API, and then we can always take additional lots, or maybe we've done a new round of formulation screening where we're pushing drug loading. We have our polymer, we're pushing drug loading. We can take, but at the same time, they're doing maybe suspension development work for GLP tox. Okay, so what we can do is we can tie in some of that suspension stability work with our new round of formulation screening. If we take our suspension and we age it for 24 hours or a week, then we can just put that into the dissolution study that we're already running and de-risk that as early as possible.
Yes, it just strikes me that you're sort of like, you're working with these companies that are very much in motion, that are like in their own external discovery processes, even as they are asking you to take on this discovery process on their behalf. And so you also have to sort of be responsive to like the changes that they're making on their end, and like that, how their drug is evolving, or like new constraints that are being applied, or like new knowledge they have about how it needs to be dosed, or like how quickly it needs to dissolve, or
right.
Like that. Yeah.
Yeah. And the dose always creeps up. Yeah, it is. It's a constant conversation. And, we love to tell the clients, the more information you can give us, the better data we can give you, the better formulation decisions we can make together. So as much as we try to be open and transparent and responsive, the best relationships are with clients that are equally as open and transparent and responsive.
Yeah, interesting. What does exploratory R&D look like from the Serán CDMO perspective? Like, are you all doing internal R&D, like in addition to the R&D that you're doing on behalf of your clients? And if so, like, what does that look like, and how does that how does that support the work that you're doing?
Yeah. So we kind of have three main branches of R&D. One is developing new technologies, right? Whether that's a new manufacturing process or an augmentation of an existing process. We also have, another branch that is, kind of continuous improvements. We have different design spaces. How can we improve them? How can we make them more robust? What sorts of new either studies can we include to de-risk things, or how can we, better design our standard experiments to give us what we're looking for. And then there's a more fundamental side. And this is where me and my group tend to fall in more, is going through all of these different compounds. And sometimes you get textbook data. You can make clear decisions. Sometimes it's a lot more ambiguous, and you start to see things that you wouldn't have expected, you wouldn't have predicted. Okay, how can we drill down into this and figure it out? So when it comes to those kinds of questions, there's kind of two approaches. One is talk to the client and say, hey, we'd love to do these experiments. We think they'd be interesting scientifically, and they could help answer some of these questions. If they're not critical path, right, then there's discussions about when and where and how. The other option is find model compounds. And this is difficult because a lot of the model compounds are, over a decade old or more. And so they're from just a different paradigm of molecules than we're seeing come through the pipeline now. We're seeing, such different approaches to the chemistry, and the targets themselves are evolving. And so it can be really hard to find a model compound that replicates the behavior that you're seeing in this client's compound. So some of it's a conversation around, like, if we were to do this for you for free, could we, include it in a poster presentation with all the data blinded? Or, if they're willing, we could publish, but, there's a lot of IP considerations around that. So that doesn't happen that much.
Yeah, that makes a lot of sense. And also, I didn't think about the fact that the model compounds are also like lagging behind the biotech ecosystem, because if you're able to synthesize a compound, you're probably focused on the patients and the use case and the actual launching, and not sort of like the modeling that's going to allow for studying manufacturability or formulation or things along that line. Am I understanding about that right?
Yeah, exactly. And just how they'll interact with the polymers is different as well. So, the kind of typical paradigm with amorphous solid dispersions is spring and parachute, right? You have your amorphous form, and you have faster dissolution, higher solubilities. So it springs up and then it crystallizes out, and your polymer's trying to just, extend that time before it precipitates out. But compounds now can completely self-sustain their own amorphous solubilities. We do still see some of the rapid crystallizers, but we see a lot of other behaviors as well. And so, those are often the ones that are more interesting, more salient from a research perspective. But those are the ones that are going to be harder to replicate.
And it strikes me that sort of like in the absence of being able to predict in advance the types of compounds that are going to be brought to you, it's hard to do exploratory R&D on those compounds. So it's sort of like the model compounds have to be sort of like in the same modality space as the modalities that are being brought to you. Am I thinking about that right?
Yeah, exactly. So the kind of how we've been approaching it, we can either try to have something that's as similar to the compounds that we're seeing as possible, or we can just have, kind of design of experiments. We're only going to vary one thing at a time. Okay, these two will control for molecular weight, these two for pKa, weak acid versus weak base, log D, right? There's all these different physicochemical properties that, if you look at enough compounds, you can start to build out an experiment to compare them all. But then you have to find the time to do more.
There's a lot of talk in the external world about AI accelerating molecule discovery, trial design, I'm assuming, drug product and formulation development, but would be interested in hearing from you, where are you using AI? Where do you see the opportunity for like AI applications in what you're doing? And like, where is sort of the external perspective on like AI automating all of this? Like you definitely like off base.
No, it's a great question. And I know it's something that we're all really interested in ourselves internally, right? Is how can we take all this data that we're generating and have it feed back into future programs? I guess I'll say the biggest limiting factor is data, right? So if we're going to use something like a neural network, right, or some sort of, Bayesian optimized model, you need tons of data, right? We're not talking about 50 data points or 500. It's like 1 million data points before you have a model that's, really fine-tuned. Even though our company's 10 years old, we've worked with hundreds of compounds, we just don't have that level of data. The other thing with data is, since we're taking all of our other clients' data, there has to be legal agreements around what we can use their data for, what we can't, right? That can also be a limiting factor. And then I think finally, the final thing is kind of this data curation that we're talking about, right? The drug substance is evolving. Our experimental methods are evolving. How we're processing it is evolving. So even though we might have data that's 10 years old, we might not be able to include it into our data sets for our compounds today. So we're really interested in it. And so there's actually, we're working on standardizing a lot of these things, not just for translatability, teachability, having new people come in and be able to learn and produce data exactly like we're doing, but also so that we can do exactly these things, is build out these data sets so we can at least do trends. Right. So if we want to do AI and ML, we need to make sure that we can trust our data. And then that's also going to guide what sorts of models we'd want to use. Right. So with this level of data that we're producing, maybe we'd be better suited for like XGBoost kind of model where, it's, and also kind of the salient part of those as well is you can see which of these, you can do much more sensitivity analysis and see, oh, wow, I didn't realize that this physicochemical property was going to be so governing of which formulation we ended up finding was the best in vivo.
Yeah, no, that's really interesting. It also brings up sort of the question of automation. Obviously, like one of the sources of the variation that you're describing is like human hands touching the experiments that you're doing. And so, interested in like where you're seeing automation being applied most effectively internally, and then where sort of the limits of where lab automation can be utilized to do the kind of work that you do.
Yeah, I mean, media prep is probably a big thing that needs to be standardized and probably could be automated. I think HPLC data workup and analysis, that's another big one that we can at least make semi-automated, if not fully. Agilent's got a nice robotic arm at its booth that can help out with that. And sync dissolutions as well. If we're thinking about it from the QC side and kind of release tests, those are much more standardized and automatable processes. There is definitely untread ground for things that can be automated. Where it stands today, I think the best we can hope for is standardization. Yeah, but I would love to see biorelevant dissolution become something that's a lot more well understood and automatable.
Yeah, that makes a lot of sense. But also, like, standardization is sort of the precursor to automation. Like in the absence of standardization, you can't automate the thing. Otherwise, you're doing something different. Do you see in the next 2 to 5 years that, generative or predictive models are going to fundamentally change the kinds of formulation science that you're doing?
Yeah, I think 2 to 5 years, it'll probably be fairly similar to what it is now as we're, building up standardized data sets. But I think that's also kind of the transition time to where we'll start to be able to include a lot of these smaller data models, right? We're already incorporating a lot more first principles models into our workflows. And that's a slippery slope to AI and ML, right? I think there's also some really cool work being done around physically informed AI models. So your loss function can include some of these first principles calculations that we're doing. And so even though it's not necessarily a first principles calculation, it can still be informed, and you can guide these generative AI models with your fundamental equations.
Yeah, and that's what I see a lot, especially from like generative protein design type companies, is they're doing a lot of that feeding back first principles physics-based models into generative models, and then, just sort of like trying to constrain it as much as possible, and then only testing when absolutely necessary, and sort of like trend that's out there.
Exactly. And I think that's you touched on something there. It is a feedback loop between the physics-based and the generative and the data. As you kickstart the cycle, things will refine, and then they're only going to keep on feeding into each other more and more.
I'm glad to hear you verified that, because that's my perception of of how it's working. You are at the intersection of so many different stakeholder groups with so many different, like, highly specific expertise, like all of which needs to feed into the decisions that are being made collectively between you and the client about like which experiments to run, how to invest the resources, how to optimize the formulation. Can you share anything that you've learned about, how to communicate across people whose expertise is chemistry or biology, to people whose expertise is engineering, to people who are more on the commercial side, to people who are on the manufacturing side, to people who, are in the lab, hands-on experiments, and may not have complete, or any stakeholders that have partial knowledge of some of these domains, but not full knowledge. It just seems like the coordination problem has to be a big one there. So I'm just curious, what lessons you've learned about how to tackle that?
Yeah. I mean, that's actually one of the aspects of this job that I didn't realize was going to be an aspect of the job, but also that I've grown to really, really enjoy. I mean, I think a big part of it is learning each other's vocabulary, right? So a chemist and an engineer might be using solubility differently versus formulations, where we're talking about crystalline versus amorphous versus colloidal versus all these different solubilities. And that really is kind of at the heart of it. Is you have to understand what they mean by the words they're saying, even if we're using the same words. And then, but I think everybody speaks risk, right? So when you're talking about, the data, where it could take you, and the current concerns that I have, putting together a table that, gives here's a decision, here's a rationale, here's a formulation choice, here's why we're choosing that. And if you can just break it down into the categories of why you're saying it and why it's important, I think that's a big thing. And then also understanding, and this is where it really comes back to building a relationship with the client, understanding what sorts of problems they've had in the past, what scar tissue do they have around different technologies or avenues that we might want to explore, because that also is going to inform how I communicate these different things to them. So I need to understand what they like, what they don't like, and how they're going to talk about it.
Yeah, it just strikes me that whenever you're sort of providing a very technical service on someone's behalf, you have to sort of understand. You mentioned the term vocabulary. You have to understand the terms under which they understand their problem before you can help them to understand their problem any more effectively. Because if you're coming at it from a completely different and disjointed, methodology and way of speaking, then even if you're giving them really good advice or really good guidance on how they should be doing their formulation, for example, they're never going to be able to take in that advice and actually understand the rationale behind it.
Exactly. And a picture is worth 1,000 words. So, I mean, we find ourselves very often turning to we're always presenting data in PowerPoint. So you can make diagrams in there, or, using something else if you need to get more fancy. But being able to just distill it down to something visual, so that then you can say, okay, here's when I say supersaturation, here's what I'm saying versus how I think you're using it. And
yeah, that's interesting. And also PowerPoint gets such a bad rap. It's scientists are always putting things in PowerPoint, but what you're sharing is that actually the reason to take it out of the spreadsheet or out of the database or out of the experiment, out of the LIMS system and into the PowerPoint, is for the purpose of translating it to your stakeholders and meeting them where they are, which may not be standardized in terms of how an AI might read it, but it is important for humanizing the data and making it consumable.
Exactly. Well, and another layer to that is we're oftentimes interfacing with part of a team, and they have to turn around and explain our data to the rest of their team. So we wouldn't be able to do that without having well-designed slides, really in-depth conversations around the data and our conclusions, and then making sure that those are captured in the slides so that they can turn around and present it. So actually, a lot of our legacy data is, you're having to go through different Excel spreadsheets and PowerPoints, figure out where it was consolidated, go back to the lab notebooks, figure out how it was collected. And we're going through right now and trying to consolidate it into machine readable forms so that we can then, and that helps you standardize the way you take future data as well as you understand what is not useful for machine readable.
Yeah. And I mean, as I'm hearing you discuss it, it also just sort of strikes me how iterative this process is, that like you learn about how your data model needs to look by the questions that people ask you and by like and slides that you need to develop, but you can't know that necessarily in advance. Like when you're running the experiment, it's just something you sort of like learn over time. And there's a feedback loop between your interface with your clients and the way you design your internal systems to capture and log that data.
Right.
And yeah,
exactly.
In terms of internal systems, what do you think is like the next stage of maturity for you all and like developing, like especially data infrastructure, but any internal systems that you feel like need to be in place for you all to be able to do your work more efficiently, more effectively, or be able to like tackle new classes of problems that clients are bringing you?
Yeah, I think electronic lab notebooks, we've made that transition almost fully in development. We're going to be transitioning throughout the company. It not only standardizes how you write your protocols, it just makes the data that much easier to go back to collect. A lot of the databases that the ELNs are stored on, you can expose them and do, SQL or NoSQL queries. And that is an immediate boon. So we've made that transition this year. I'm really excited to start digging into the data.
Can you share a little bit about like what that transition was like? Like what were the hardest parts of transitioning into ELNs? And I don't know, maybe before what the hardest parts, like how did you all decide that it was time? And then, after you took it on, like what were the hardest parts of making it happen?
Yeah. So we decided to take it on, I think, out of this, it helps with review, it helps with standardization. So those are really the catalyzing factors, but we also have people at our company that have helped set up ELNs or came from labs that only used ELNs. And so there's just already a lot of interest in it. So we've been working on it for a while. The hardest part is building out the protocols, especially in a development environment where we are tailoring our experiments in so many ways. And we have such variable numbers of samples that can go into it and so many different experiments and unit operations that we're running. Building out all of those templates takes a really long time, and it's a lot of working with the ELN company to try to see, what they already have kind of templated, what you can adapt of those, what you need to write from scratch. And then there's a lot of learning how to write them from scratch. So I would say writing the protocols in a way that fits our system is the majority of the time that it's taken.
What is the hard part about writing the protocols? Is it translating it into the ELN can understand, or is it, documenting things that have never been documented before? Is it like, hashing out among each other what the protocol should actually be versus what we've actually been doing on something else? I don't know.
A lot of it is edge cases, right? So you start out by building your protocol for the standard, okay, it's a biorelevant to solution, 5 samples, N of 2. We're going, from pH 1.6 to 6.5 of biosalts. Okay, but what if we're doing a biorelevant to solution and trying to de-risk food effects, or we're trying to understand, drug-drug interactions where the patient population is going to be taking proton pump inhibitors, or maybe they have a physiological, achlorhydria where their stomach is going to be, higher. Their stomach pH is going to be higher. So the media, right there, you're having to change or develop additional recipes. Okay, well, what if we want to only do a 1-stage dissolution? Or what if we want to do a 3-stage dissolution, either because we're doing a colonic pH, or because we want to understand how our undissolved solids or precipitated solids are going to, redissolve and be a little bit more, get some insight into what's going on in the intestine that way. And that's just disso, right? And then there's manufacturing processes of, okay, how many different formulations do we need to produce? How many samples do we need to take? What types of samples? Where are they going to go? How do we track all of these? Is a stability sample the same thing as a dissolution sample? So it really is just like taking all of these standardized Word doc protocols that we adapt to every program, to every step of the way, and trying to get a single document or protocol that captures at least 90% of those, and then being okay with going back and building specialized ones.
Right. So it's just like an extension of all the different questions that you might ask times all the different permutations of like of the ways that you might ask it, and you end up with just an endless list of protocols. As we head toward the end, as you think about your experience, like so many organizations, I mean, like Serán, you're a product sort of your history, and like the way that you grow and develop, the clients that you take on early on, and like the methods that you've developed, and all of that. I'm interested in it. If you were designing like a data first CDMO from scratch, like are there lessons learned or like ways that you would think about it differently if you were starting again?
I think starting with an ELN is the big thing. And then that way, I think another important thing is you can track how your experiments evolve. Because if experiments are going to evolve, science is evolving, the models are evolving. And so how you collect the data is going to change over time. But if you have it in that way, in an ELN, everything is going to be tracked. I think that's the big thing, is thinking about how you're storing your data. File tree structure. That's another big one. Oftentimes those evolve organically, but being able to, because if you can build something to crawl through your file tree and pull out your different bits of data, that would be huge too. I think those are kind of the two big ones.
Where can people go to learn about you and the work you do at Serán?
We have a learning center on the Serán Bio webpage. So all of our white papers, any of our presentations, so you can listen to them all. And then any white papers are going to be there as well.
Yeah, well, awesome. It's been great to see you here at AAPS Pharmacy 360. Really appreciate the time and look forward to connecting down the way.
Yeah, thanks for having me.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.